So now we'll survey the male reproductive system, beginning with the scrotum and testes and continuing through the spermatic ducts and accessory glands that are associated with them. So the male external genitalia consists of the scrotum and the penis. They occupy the perineal area, which is that diamond-shaped area between the thighs, bordered by the pubic symphysis, ischial tuberosities, and coccyx. The scrotum itself is a pouch of skin and muscle and fibrous connective tissue that contains the testes. The left testes usually descends a little bit lower than the right so that the two are not compressed against each other and for dissipation of heat. The skin over the scrotum has sebaceous glands, sparse hair, very rich sensory innervation, and a little bit darker pigmentation than the skin elsewhere. There's a median septum that divides the scrotum into the right and left compartments. And the seam of that is called the perineal raphe. It's located anteriorly along the ventral side of the penis and posteriorly to the anus. The testes are suspended in the scrotum by the spermatic cord. The spermatic cord is a bundle of connective tissue that contains the ductus deferens, blood vessels, lymphatic vessels, and the testicular nerve. It goes through about 4 centimeters of inguinal canal as it ascends into the pelvic cavity. It's bordered on the inferior entrance by the external inguinal ring and on the superior exit that goes into the pelvic cavity by the internal inguinal ring. And the whole purpose of the scrotum is to suspend the testes outside of the body because they've adapted to generate sperm in a much cooler environment, about 35 degrees C, relative to core body temperature of 37 degrees C. So you can see the spermatic cord here, consisting of the testicular arteries, the pampiniform plexus of veins, the testicular artery, and in there's the testicular nerve also. So it's very important for the scrotum to maintain a cooler temperature than the body. And it does this through 3 mechanisms. The first of these is the cremaster muscle, which results from strips of internal abdominal oblique muscle that descend down through the inguinal canal. You can see that here, covering the spermatic cord. In cold temperatures, it contracts and draws the testes upward towards the body, whereas in warm temperatures, it relaxes and suspends the testes further down away from the body. The second mechanism is the dartos muscle. The dartos muscle is a subcutaneous layer of smooth muscle, and it contracts when it's cold, causing wrinkling in the scrotum. It holds the testes closer to the warm body. It also reduces the surface area of the scrotum and thus reduces heat loss. The third and final mechanism is the pampiniform plexus, which is an extensive network of veins from the testes. It surrounds the testicular artery and the spermatic cord. It's a countercurrent heat exchanger. So this figure here shows as warm blood from the body descends through the artery into the testes, it gives off heat to the venous blood returning to the body. You'll notice the arrangement here in the testes. This pampiniform plexus is highly branched and it's external to the arteries. Thus, it aids in removing heat from the blood so that cooler blood is entering the testes. Without the pampiniform plexus, this warm arterial blood would heat the testes and that would inhibit sperm production. So the testes are both endocrine and exocrine glands. That is, they produce sex hormones, which are secreted into the blood, and then they are exocrine in the fact that they produce sperm, which is released into a duct. The testes are oval and slightly flattened. They're about 4 centimeters long and 2.5 centimeters in diameter. They're covered anteriorly and laterally by this layer called the tunica vaginalis. This is basically the internal portion of the sac into which they descend. The tunica albuginea is this white fibrous capsule on the testes. If we were to remove the tunica vaginalis, we would be able to see that. The connective tissue septa is going to divide the testes into 250 or 300 different wedge-shaped lobules. Inside these lobules, we will see the seminiferous tubules. Let's take a closer look at seminiferous tubules. There's 1 to 3 of them in each lobule. They're very convoluted and lined with a thick germinal epithelium for sperm generation. We have interstitial or Leydig cells that are between the tubules, and those produce testosterone. And then there are sustentacular cells or Sertoli cells that we'll see inside the tubules, but between the germ cells. Here you can see in a scanning electron micrograph that we have the tails of the sperm in the center of the tubule, and we have the Sertoli cells here in purple, and the germ cells developing sperm in green. The Sertoli cells, or the sustentacular cells, protect the germ cells and promote their development because these germ cells need to develop after meiosis into fully mature sperm by the process of spermiogenesis. The germ cells depend on the Sertoli cells for nutrients, for waste removal, for growth factors, and several other needs. The sustentacular cells form the blood-testes barrier. Now, it's important to have a blood-testes barrier because the germ cells are immunologically different from the body cells. They have different genetic makeup, after all, and they would get attacked by the immune system. So the sustentacular cells, or Sertoli cells, separate the sperm from the immune system by providing this barrier. So a sustentacular cell is shaped like a little tree whose roots spread out across the basement membrane of the tubule, forming a boundary. The thick trunk reaches into the tubule lumen, and there are tight junctions between adjacent sustentacular cells, which form the blood-testes barrier. So the seminiferous tubules from each of these lobules will dump their contents into a network of tubules, the rete testes. A point to note is that sperm don't swim while they're in the male reproductive tract. They don't have that ability yet. The testicular artery supplies each testes. They have very low blood pressure, in fact, so low that there's not even a pulse. This creates a very low supply of oxygen in the testes, and that causes sperm to develop very large mitochondria, which helps them survive in a more hypoxic environment, like that of the female reproductive tract. Blood's going to leave the testes, as we've examined, through the pampiniform plexus, which converges to form the testicular veins. The right testicular vein drains into the inferior vena cava, and the left one drains into the left renal vein. The testicular nerves come from spinal cord segments T10 to T11, so the thoracic region, and they carry sensory fibers that are concerned with pain and motor fibers that are concerned with regulating blood flow. After leaving the testes, sperm will travel through a series of spermatic ducts to reach their urethra. So we've already seen that the seminiferous tubules dump into the rete testes. After dumping into the rete testes, they will enter the efferent ductules. These are about 12 small ciliated ducts, so little hairs on the inside, that collect the sperm and transport them to the epididymis. The epididymis, or epididymes for plural, otherwise known as the duct of the epididymis, is a very long coiled duct. It's about 6 meters long. And as it coils up, it forms the epididymis, which is about 7.5 centimeters long. It has a head region, a body region, and a tail region. Now, it'll take the sperm about 20 days to travel through the entire length of the 6-meter long duct of the epididymis. They mature as they travel through the duct, and they remain fertile for about 40 to 60 days. And if they're not ejaculated, they're going to disintegrate, and the epididymis will reabsorb them. So one of the major functions for the epididymis is for maturation and storage of the sperm. After the sperm reach the tail of the epididymis, they'll move into the ductus deferens or the vas deferens. The vas deferens is a long muscular tubule. It's about 45 centimeters long, and it passes up from the scrotum through the inguinal canal to the posterior surface of the urinary bladder. There, it's going to descend down into the terminal ampulla behind the bladder. The duct ends by uniting with the duct of the seminal vesicle, and it'll dump into the ejaculatory duct. This thick-walled ductus deferens is well innervated by sympathetic nerve fibers. The ejaculatory duct that we see here passing through the prostate gland is only about 2 centimeters long. It empties into the urethra. And the urethra, as we know, is shared by the reproductive and urinary systems. It'll extend from the ejaculatory duct all the way down through the penis. Consists of 3 regions: the prostatic, the membranous, and the spongy urethra or penile urethra. So as the spermatic ducts make their way from the testes to the exit of the penis, there are 3 sets of glands that they'll encounter. First, the seminal vesicles, which are a pair of glands posterior to the bladder that we see here. Each is associated with 1 ejaculatory duct, and they produce a yellowish substance that constitutes about 60% of the semen. Second, we'll pass the prostate gland, located right here. It surrounds the urethra and the ejaculatory duct, just inferior to the bladder. It has about 30 to 50 compound tubuloacinar glands, and they empty through about 20 pores into the prostatic urethra. They have a thin, milky secretion that constitutes about 30% of the semen. Finally, we'll see the bulbourethral gland right here, or the Cowper's gland. It's named because it's near the bulb of the penis. During sexual arousal, they produce a clear cyphery fluid that lubricates the head of the penis in preparation for intercourse. But more importantly, probably, it protects the sperm by neutralizing the acidity of residual urine that may have been in the urethra. That brings us on to the penis. The penis serves to deposit semen in the vagina. Half of the penis is an internal root, and half is externally visible as the shaft. The external portion is about 4 inches long when flaccid, 5 to 7 inches when erect. The skin over the shaft is loosely attached, which allows great expansion. It extends over the gland as the prepuce or foreskin, unless that's removed by circumcision. Schmegma is a waxy secretion produced by the sebaceous glands in the glands and facing the surface of the prepuce. There are 3 cylindrical bodies of erectile tissue within the penis, which fill with blood during sexual arousal and account for its enlargement during erection. There's a single corpus spongiosum along the ventral side, right here. It encloses the spongy urethra, right here. Its distal end enlarges and forms the glans penis. The proximal end forms a dilated bulb, which is ensheathed by bulbospongiosus muscle. There are 2 corpora cavernosa. They diverge like arms of a Y. Each arm is called a crus, attaches the penis to the pubic arch, and it's covered with ischiocavernosus muscle. So that concludes our section on male reproductive anatomy. Shortly we'll move on to the next section of this chapter, puberty and the climacteric. Before you do that, go out to Anatomy and Physiology Revealed and watch the animation that I've assigned that gives you a really nice overview of the male reproductive system. Also, take a moment to put together a little diagram. Consider the anatomy of the scrotum and its contents in the testes. Look at the dartos, the cremaster, the seminiferous tubules, how the pampiniform plexus works, the rete testes, the efferent ductules, the duct of the epididymis, vas deferens to ejaculatory duct, to urethra. And then put the glands in place, the seminal vesicles, the prostate, and the bulbourethral gland. Spend a few minutes while it's fresh in your mind and get a picture of the content that we just covered.